Ladies and gentlemen, welcome to this latest teaching session on the course on technology and the future of medicine. Today, Ross Lockwood is going to take us through the promise and perils of nanotechnology. Take it away, Ross.
Thank you very much. So, as I mentioned, this lecture was previously prepared by Dr. Michael Woodside, and I'm giving it him sort of on his behalf, but it's been so many times now that it's at this point, this will probably be the last time I give this presentation, and I intend to make my own of the same version. But we're going to kind of follow along the same theme as my last lecture when we were talking about the kind of history, a very brief history of digital computing and silicon technology in the context of nanotechnology. So we were looking at how we were manipulating atoms and electron orbitals to do computations. And as you'll see today, in Michael's original lecture, he actually does talk about some of the things that we talked about last lecture, but he goes on into some of the more biological applications and medical applications of nanotechnology. So this is the promise and perils of nanotechnology. When we're talking about nanotechnology, we have to come up with some definitions for what exactly it is. Nanotechnology itself is a very diverse field, and it typically is considered to be at the intersection of physics, chemistry, biology, and engineering. Because when we talk about nanotechnology and we talk about nanoscale objects, we're talking about things that have existed for, the length of the universe, but we humans are now understanding how to manipulate those things. And we use the understanding that we have from biology and from chemistry and from physics and from engineering to do those things. So examples of nanotechnology in progress today, we can do things like build structures by manipulating individual atoms. This is like dragging atoms along a surface with a scanning tunneling microscope or equivalent device. So things that fall under this category: quantum dots, atomic corrals, and perhaps even new architectures for computation. And then another approach is to use biology as your template for nanotechnology. Because a lot of the things that we're talking about in terms of what we want to make on the nanoscale already exist in biological examples. So biomotors are a good example of that, but there's also things like DNA origami, or something that I did a couple of months ago, a workshop on synthetic biology. So we'll touch on all those topics today. So the first one, we've kind of already seen this, is manipulating atoms using a scanning tunneling microscope. So this is a microscope that has a tip that's connected through a circuit to the sample itself, and it just measures the current as a function of distance. So when it gets close enough, it actually leverages a quantum mechanical effect called tunneling, hence why scanning tunneling microscope, why tunneling is in the name. But once it reaches a specific tunneling current, then it will scan across the top of the surface, maintaining a constant height. And those heights that it reads out are the actual data that are displayed on screen. So if you're looking at a surface of atoms like this, the tip is actually following the profile of the electron orbitals that are accessible to the tip. So it's basically as close as you can get to the surface without touching it. You can get closer, but that being said. So this is an image that's produced by this. This is a silicon, I think it's a 110 surface. So the silicons appear bright, and of course, because of the way the crystal lattice is arranged, you actually get things like gaps and these beautiful patterns as a result of the crystal orientation. So Eigler et al., they leveraged an STM to actually be able to write. So this is actually better than we were talking about with Feynman. We were talking about 100 nanometer domains. Now we're talking about domains the size of atoms themselves. And it's very difficult to see, I think. But in that bottom right-hand corner, it's actually the word IBM being spelled out in atoms. And I think that was on a copper surface with something like gold atoms. But I'll have to check. And you can do funny things like this. So here's a little stick man made out of atoms. And IBM has actually made an animation of this and called it the world's smallest movie. But one of the really interesting things about being able to manipulate atoms is that as you're manipulating the atoms, you're also manipulating their quantum mechanical properties. So structures like these quantum corrals have emerged where if you arrange atoms on a surface into a particular structure, you can actually create interference of electron waves to produce artificial maxima So in the case of the bottom right-hand corner where we have this kind of perfectly circular corral, you can see there's a wave on the interior of the corral. And this wave is not an atom, there's not something there, but it's the interference of all the electron wave functions from the atoms in the corral interfering to form a maximum in the center. And as a result, you actually have properties that are atom-like at the center of a corral like this. So remember at the end of the my last lecture, we were talking about what kind of things can you do with quantum mechanics? Well, you can change material properties. So in this case, the corral may actually have different properties than you would imagine if the electrons weren't causing that interference themselves, or if it was in some disordered array. Okay, and there's lots of things that you can do with this. So here you go, ripples from the electron waves are forming that central maximum. So you can do different shapes. You can create different field profiles on the interior of these corrals. But one of the most interesting things is that you can actually create images of atoms. So here the corrals are ellipsoids, and they work kind of the same way that concave mirrors work, that there's a focal point, and if you had another concave mirror attached to it, you would form an image at the opposing focal point. So in the bottom screen, you can actually see on the left-hand image anyway, here's the actual atom that's at one of the focuses, and then on the right-hand side, there's an image of the atom. So these are all just examples of ways that you can manipulate the electronic properties without actually introducing any materials. All you're doing is changing the geometry, and you're producing electron wave functions that are different as a result. So here are some other examples. When the when the atom is not at the focal point, you don't get this interference pattern, so you don't see the image. So this might be a state machine that can measure zeros and ones, whether or not there's an atom at the focal point or not. But nonetheless, you can see that there are many material properties that are just emerging as a result of us being able to manipulate atoms. So while this is a powerful tool for science, it's difficult to bring this into the consumer space, to bring this into the commercial space. Because, so first of all, it's a direct mechanical manipulation of individual atoms is a remarkable achievement. But it's still very technically challenging. So you need to have very stable machines. So if you go over to NINT and you do a tour there, they'll tell you about how their concrete floor is detached from the building so that any machines that are on the concrete floor don't suffer from the vibrations in the building. Because you can have things as simple as someone walking on the floor creating enough vibration in the underlying structure of the building that gets these machines out of tune and out of whack and gives them poor readings. So you need to have very stable environments. A lot of these machines operate at very low temperatures, so either liquid nitrogen temperatures or the more expensive one at liquid helium temperatures. And as a result of these two things, you're very constrained to the kind of things that you can study. So it's not very easy to study, say, organics that are on these types of surfaces, although people do it, and there have been some breakthroughs in the last 5 years that are pretty impressive. So like direct imaging of a chemical reaction and bond formation is one example of something that's been done. But it's very difficult, and there's still high error rates. So the real trick with this, if you're going to see this as a nanotechnology, is to try and scale it up. You can't just build one of these machines, right? You're going to have thousands of copies of them, and they're going to have to be cheap enough that, if they break, they can be repaired quite easily without specialty knowledge. So while it's a powerful tool, it's not yet mainstream. And even after 20 years of research, it's not quite routine. But now we'll go back just to one of the examples of things that you can do with this machine that may become relevant in the next 5 to 10 years. So here again is the silicon surface terminated with hydrogen atoms. And in this little animation, it kind of illustrates how you can give a little electric charge to one of the hydrogen atoms and have that hydrogen leave the surface and leave behind a dangling bond defect. So these white spots here are where there should be a hydrogen, but because there's no capping hydrogen, an electron orbital sticks out and has different electronic properties than the surrounding surface. So this is just being able to draw a straight line of these. But what happens when you can do a pattern? Well, a pattern like this one allows you to create basically a state machine and create circuits as a result. If you have 4 of these silicon dangling bonds, you can actually do things like change the charge distribution around them to create different states. So here we've added charges on the corners, and you can see that the electron distribution of the actual 4-cell unit has changed. So you can imagine something like that being incorporated into the wire. So you refer back to the movie about the electrons swapping seats and chairs, because I think that that's here. But where this becomes really powerful is when you combine it with this quantum dot cellular automata. So cellular automata is basically you have a grid of cells with discrete states. So you can imagine that these are the hydride bonds themselves, whether they're polarized in one direction or the other. And they evolve by sampling the states around them. So there's some environmental awareness to this. And so a very famous version of this is called Conway's Game of Life. And you can create some very interesting structures, like this one is creating glider guns as it progresses. So as it moves to the right across the screen, you actually get these little glider guns shooting gliders out on the top of the horizontal axis there. But there's all sorts of other things. And in fact, they have demonstrated that Conway's Game of Life itself is Turing complete. So you could actually create a computer using nothing but Conway's Game of Life, the universe of Conway's Game of Life. So in this example, instead of having black or white cells or living or dead cells, we have binary ones and binary zeros depending on the electron distribution. So you can create things like wires where the state at one end is the same as the state at the other. More importantly, you can do things like inverters, where the state at one end is the opposite of the state at the other end. And then, of course, you can do things like fan out, so you can split signals into many other signals. And these three structures and many other structures like it would form the basis of just such a computer. So you can imagine and, or, not, exclusive or, all these different gates can be formulated in the language of this quantum dot cellular automata. So that basically is what we were talking about last time in terms of the history of digital computing on a silicon surface. But now let's talk about some nanotechnology that has more relevance in the medical space and the biological space. Now, to give you an example, here are basically the building blocks of, well, using biology as a template. So instead of looking at physics and looking at cellular automata, looking at nature itself and seeing, what can we do with the things that are here. And DNA is one of these structures that is very interesting and very easy to work with. And now with all the tools in our genetics toolbox, we can actually do genetic manipulation of DNA. We can create different strands with different amounts of code. And you can actually create structures like these happy faces in this, the cover of this Nature magazine entitled DNA Origami. So the way that this works is basically using the properties of the DNA itself, how the base pairs interact with one another, and you create a strand of DNA that is ordered in such a way that every once in a while you'll get different parts of the DNA strand snapping together and binding with themselves. So in this way, they developed basically a method of producing a bunch of different shapes. Now, I looked at this when I first saw it and I was like, oh, whoop-de-doo, a bunch of shapes. Now keep in mind that when I saw it, it had just come out in 2006 and I was doing a second-year paper on nanotechnology at the time. But what I didn't fundamentally understand is that all functionality that we have in the real world comes from being able to manipulate the geometry of things. So while we have things like catalysts that are proteins that help different chemical reactions move forward, the shape of things is really what does a lot of the work, okay? I'd say one of the major components of how you interact with something is what shape it's in and what it's designed to do. Like a knife has a very sharp edge, right? And it's important to the knife that the geometry is maintained. So here's an example of people that are taking DNA and creating these very cool structures. So, of course, the next question is, well, what is some of the useful stuff that you can do with this? So by engineering these things, you can do what we were discussing before. You can write things in DNA. You can have like a little tapestry, little maps. And then you can create these structures that are standalone. But because they're engineered in a particular way, each side of the surfaces has a different way of connecting. And so you can actually get self-assembled structures like these big hexagons and some of these triangular patterns, where all you did was create the DNA strand. And all you did was let it fold up. But what it did after the fact was get together with a bunch of neighbors and make some larger structures. So already we're going from something that's the size of a DNA strand, and we're able to build structures that are larger. And the important part is that it's self-assembly, that there's no action on the part of the human other than maybe changing the temperature here and there and jiggling the bathwater a little bit. But you can create these larger structures. Now here's a really cool application. Suppose you've done this and you've created a six-sided object. Well, you've basically designed a little box. And what the group did with this particular box is they created a little strand of DNA that acted as the lock of the box. So that when the complementary pair arrived, it unlocked the box, and then whatever was in the box was released. So what's really interesting about this is that you can immediately sense applications for drug delivery. You can put a drug inside of the box, close the lid, and lock it by a chemical mechanism. And you can make it so that the unlock for the box is something like a tumor cell or an antigen that's present in a particular tissue. And in that way, you can have the patient take a whole bunch of these origami-style drug delivery things, and the drugs will be delivered to the specific spots where they can be unlocked in the body. So that's one potential use of just such a structure. Now, of course, that's talking more on the biological sides, but DNA is robust in terms of its material strength as well. So if you're considering building larger structures, still on the nanoscale, but larger structures, you can make scaffolding platforms, you can make lockable interconnects, you can make all sorts of interesting things. And again, they kind of do the same thing as we were discussing before, is that they tend to self-assemble in ways that have been programmed, designed into the experiment itself. So even three-dimensional structures like these dodecahedrons on the bottom right-hand side are possible with this type of technology. So this is really one of the first attempts at starting with, or at doing basically ground-up nanotechnology, where you start with something small and then you create something big. Now the scaffolding here is all the genetic history that we now know about that basically emerged in the last 30 years. But nonetheless, even though we're using a pre-existing structure like DNA, you can still do a tremendous amount of things with it. So here's some other examples. Some DNA origami was used to create artificial membrane channels. So you can use this as an antibiotic for DNA sequencing. If you could somehow thread a strand of DNA through the central channel of this thing, you could actually measure potential differences as different base pairs. But fundamentally, the powerful methods here is that you can create complex structures using very simple ingredients, and you have a very high throughput because they tend to self-assemble. The trouble there is that it's a very limited repertoire of materials, so we're limited to the 4 base pairs, or if you get into debates online, it could be as many as 6 or 8 or a little bit more than that. There's still some chemical compatibility in terms of turning this into more functional materials, but nonetheless, it represents a good foundation. It is useful in a limited range of environments, so when we're talking about using biological, or starting with biological structures as our building blocks, we're already limiting ourselves to basically the range of temperatures and conditions where life can exist, right? You start to talk about cell damage if you're getting too hot or too cold, and things like this. And then the one at the bottom there, that there's no error correction, has a question mark because DNA fundamentally does have error correcting mechanisms. It's just a matter of leveraging the cellular structures that already do it on your applications. And so that's still a question that is not fully answered, but hopefully in the next little while it will be. So in terms of biology, we already have a number of very interesting structures that we can look at. So muscles themselves are linear actuators. You can use a muscle to pull on a strand of actin, right, using a little myosin, head of myosin. So here's a little animation showing how this kind of ratcheting style action works in this myosin head walking along an actin filament. So that's how our muscles contract. And you can imagine leveraging that on the nanoscale. If you needed to have something that pulled on a linear scale, you could leverage a structure like this. More complicated are motor proteins. So we've got the kinesin molecule walking across a microtubule. Basically what you see in this image is a long microtubule and a vesicle that's being pulled along the microtubule by a kinesin molecule. So if this animation had worked, you'd see this little dot moving very slowly along this microtubule. So this is another hint at how do you move things within nanostructures from one place to another. You can have microtubule highways and myosin molecules that are basically their only job is to pull things along these motors, or along these filaments. And what's really interesting is that, on the biology side of things, people have been studying these molecules, trying to understand how they work. And myosin walks along these filaments with hand-over-hand motions. What these graphs tell you is what the size of the step is. So if the step size is larger than 1 pace, if it's 2 paces long, then you know that it's walking hand over hand or foot over foot, instead of doing the shuffle where you're just matching up 1 foot at a time like this. So, and that depends on what your application is going to be. So maybe you want to design a molecule that doesn't do hand over hand motion. How would you do that? This isn't Michael Woodside's research himself, but he does very similar things. So you can actually monitor the motion of a single molecule using something called an optical trap. And an optical trap works thusly. Basically, you take a little bead, a couple of micrometers across, and you trap it in the focus of a powerful laser. So the shape of the beam actually pulls the bead back into the center of the optical trap. So now you have very fine control over where that bead is in space. And what they've done is they've taken a kinesin molecule and attached it to the bead and then presented it with a microtubule. So basically now you've got the laser holding onto the microtubule and the kinesin doing its thing down on the microtubule, trying to walk. And what they're measuring is the position versus as a function of time. So they're actually the laser is letting the bead move along this molecule, and you can see that the kinesin is taking little steps. It has these dwell periods where it's being reset or waiting for ATP to come and supply it with more energy to continue its journey across the microtubule. So that's an example again of a linear motor. We do have good examples of rotary motors. So one of the most famous ones is the ATP synthase motor. And this one functions as a rotary motor for hydrogen gradients, and it drives rotation to create enzymatic activity of the alpha and beta subunits. So the studies done on this molecule were awarded the Nobel Prize in 1997. And basically what they discovered is exactly, or what was discovered is exactly how these motors rotate. So in what step is energy being used, in what step are products being sent away. This fluorescent actin is the same as the little bright spot here, and what they found is that you can actually watch as this filament is basically tossed around, is rotated around the motor protein. So here's a little graph of showing where exactly that fluorescent actin filament stops. And you can see that it's taking 3 discrete steps as it's progressing around. A similar motor, the Flagella motor, is another object of study. And this one works by having basically a hook attached to a rotary motor protein and then a propeller behind the organism. And so you can actually just imagine that this thing makes a little corkscrew pattern with the filament, and that drives the organism forward. Now, one thing to remember is that when we're looking at pictures like this, we're looking at actual molecules. So if you imagine water is a pretty small molecule, it might not show up very well in an image like that, but we tend to imagine it without the water present. So if you imagine that water's there, this filament, this propeller behind it, isn't acting on liquid water in the sense that we like paddle through water when we're swimming, but it's actually interacting with individual molecules and kind of pushing them out. So it's more like, imagine yourself in a ball pit and you have one of these motor proteins, that your propulsion through the ball pit has more to do with how you're pushing the balls behind you than it is how you're swimming through a liquid. So that's the kind of thing that you have to be aware of when we're down at these scales, that things like water don't behave the way that you expect them, and things like air have different properties, right? To take into consideration if your machine, say, is susceptible to oxidation. So the studies that were done on these things basically used fluorescent beads to track the position of the flagellum. And depending on kind of the length of the flagellum and the conditions of the environment around them, you actually get different rotary properties. So each of these little fluorescent beings in here is Listeria. So it's Listeria that has been dyed with a fluorescent label. And basically what you see is the Listeria are propelling themselves by a different method, but they shoot around these cells. Listeria is the bacteria that causes food poisoning. And the way that they're doing it is actually by catalyzing an actin filament behind themselves. So they actually cause the cell's machinery to misfire, and it propels the bacteria itself around. So basically, it hijacks the cell's machinery to make actin, and then leaves behind a trail of this actin garbage as it propels itself through the cell. So it's a very interesting method of producing motion, but nonetheless creates motion on the nanoscale. So it's one of those things that you could conceivably engineer. So here's just kind of an example of how this actin propulsion works. Basically, every time a monomer of actin is polymerized behind the bacteria, it pushes the bacteria forward by a small amount. And in this way, it ratchets forward through Brownian motion and this polymerization. When we're talking about actually creating things on the nanoscale, there's a couple of things that we need to do. Being able to transport molecules is one of the primary ones. So let's say you've got, a nanotechnology unit that you want to extract gold particles from sewage water. That was a big thing in the news this week. Is there's millions of tons of gold, or not millions of tons, millions of dollars worth of gold that is just in very low concentrations in sewage water. So how would you imagine extracting that very small quantity of gold out? Well, you might even consider going as small as a few atoms of gold or a collection of atoms of gold, but nonetheless, you're going to have these motor proteins to move it to a specific place. So capturing, transporting, and then in these ones, they have additional steps: tagging, transporting, and detecting. So knowing when you have something is also an important problem to solve. And many people have gone ahead and just gone and designed their own motors. So here is an engineered motor by Kuwata et al. In Physical Review E, 2011. So they have these repressor protein feet. They've got ankle joints, hub joints, and coiled legs. Presumably, this basically rotates as it walks down a DNA track. So rather than using existing cell structures, they've designed something that can walk along a DNA track. And this is where things get interesting. Synthetic biology is a very exciting topic in the field because basically anybody can do it. This December, I went to a synthetic biology workshop where they basically walk you through creating a bacteria that will read a customized plasmid with the proteins that you wanted to express. So in my case, I wanted to express red fluorescent protein and blue fluorescent protein in the same bacteria. Basically what synthetic biology does is it gives you the building blocks to be able to create these DNA plasmids. So for just to give you an example of what I did, basically you take this anchor for the plasmid. So I'm assembling a plasmid with these synthetic circuits. So basically what people have done is they've found a series of parts that you can make and copy very easily using standard genetic tools. So someone in the field produces a little anchor that they want to use for the beginning of a plasmid. And then they go ahead and make copies of it, and then they sell copies of it. They say, I'm the guy that makes the anchor for this DNA plasmid, by whatever means, but they can make as many copies as they like using the standard genetic tools. And then other people say, well, I made a protein for green fluorescent protein, and I can connect to your anchor because I have this sequence of DNA at one end of it, or RNA, or whatever you're using. And basically, you take anchor, you snap it to the green fluorescent protein. You want to do something else, you snap it to the end of the green fluorescent protein. At the very end of your plasmid chain, you have another DNA sequence. That's your cap. And once the cap is assembled, it will actually close the plasmid loop, lock it in place, and then you just do a simple process to put the plasmid into the bacteria, and the bacteria will start expressing those proteins. So this is something that you can buy a kit for less than $100, and you can have your own synthetic bacteria at the end of the evening. It was a 3-day course. But here's an example of what you can do with it. So E. coli here are engineered to be photosensitive, so changing their color depending on the amount of light that's present. And it may be difficult for you to see, but there's a portrait there. of the author in this Nature paper. And then you can also imagine, and this is something that's been done, that you could create your own rules for how an organism works, your own metabolic pathways, your own protein expression. And in fact, in 2010 it was done, a bacterium with an artificial chemically synthesized genome was created. And that was Gibson et al. In Science. So this opens doors for creating your own bacteria that have very specific functions that work in a very narrow range of environments. So the danger here, of course, is that by creating synthetic biology, you may introduce it into the real world where it competes with regular bacteria. And if it ends up that the synthetic biology that you created is dangerous to humans or plants or animals or any existing organisms, it's going to compete with them with the same fervor that we compete with the rest of life. So that's one of the downsides there. The applications for synthetic biology, obviously biosensing, being able to create bacteria that detect particular things and change to particular colors. So perhaps an E. coli bacteria that detects when your chicken has gone bad by coloring it blue, right? You wouldn't want to eat a blue chicken, neither would I, so that, is a great example. Producing biomaterials, so ethanol production, being able to do big harvests of biofuels and pharmaceuticals. Of course, cellular digital computation is also on the table. So just imagine in the example of the silicon surface that instead you're replacing the computing elements there with, say, the DNA origami, so that if it's got two neighbors, it will snap together to form a structure that represents a state, a 0 or a 1. And this also has been demonstrated in recent years, and it's a very powerful technique at sampling a very large parameter space, so solving problems that have a lot of different parameters. Creating synthetic pathways or systems to test disease mechanisms and therapies, so we can introduce, say, a new metabolic pathway that can sample, the progression of cancer in a patient or something like this, that tells you basically how far along certain health-related metrics are. We can engineer viruses as therapeutic agents, so targeting drug-resistant pathogens, increasing antibiotic efficiency. And any of the tools like that obviously could be repurposed for treating cancer, so creating a tumor-invading bacteria, which also delivers drugs, and modifying disease vectors so that they are rendered harmless. So creating genetic variants of mosquitoes has been done in the past, where the females themselves become flightless and are less available for mating, highly reduces the population of mosquitoes. So while there is a wealth of tools available to synthetic biology, Michael Woodside likes this quote to describe kind of the state of the field right now, and it's that the genome is difficult to read. So bought the book, hard to read, right? We've done the Human Genome Project in the last 10 years, and we're on to the next kind of generation of these genome projects. But at the moment, it's not so simple for you to get your own genome sequenced. Although there are products, and I'd be happy to tell you about my experience with 23andMe, where I sent a spit sample to them, and they analyzed it for a bunch of common genetic factors. So basically, they told me about some of the health history that that my family and family tree have had, that I can expect maybe later on in life as they develop. And then, of course, synthesis. You can write in DNA, but you have little to say at the moment. So on both ends, we kind of understand how synthetic biology will play a role in the future of these things, but the tools on both ends need to be better developed. So being able to speak the language of DNA a little bit better, and of course, once you're able to read that, being able to write your own that does something in a pre-programmed way, so the synthesis part. And this is Michael's research himself. So what he tends to do is he'll take one of these bead experiments where he's got these focused lasers, laser manipulators, and he attaches onto them DNA anchors that then latch on to a particular protein. And this is a very interesting problem because protein misfolding is a big factor in diseases like Alzheimer's. Basically what he does then is with these trapped beads is he pulls very gently on the ends of the protein, and he'll know precisely what force is available or is necessary to pull the protein and unfold it back into a linear chain. So basically you get these folding and unfolding graphs that tell how difficult it was to pull on the protein until the next unfolding event. And The tractor beam is basically the way to describe how these beads are being held inside of these lasers. So the lasers and the beads, the lasers are basically little tractor beams that you can pull these beads around wherever you like. Rather than a bead, they've trapped a bacterium in the focus of this laser. And in this image, the bacteria is struggling furiously to get out of this tractor beam, but they're just basically moving it to wherever they please inside the sample. So you actually get this very comedic kind of, I never laugh so hard at the misfortune of a bacteria, but here's a great example of something. So I'll try and look that up after the class here and email it out to you guys, swimming E. coli bacteria. Other protein diseases, prion diseases, the mad cow disease, Scrapie, Creutzfeldt-Jacobs disease, they're caused by a prion protein, PRP. And so basically this is a protein that misfolds into an infectious structure. So every once in a while this protein is malformed and folds into an infectious structure. But in doing so, it actually catalyzes more of the infectious protein to be formed as well. So you have this runaway effect where as soon as you've got one of them, you start proliferating more and more of them. So how does this operate as a catalyst? How does it convert native molecules to the infectious molecules? What is that infectious structure? And how specifically does it kill neurons? So this is, while we're saying that these are diseases that we're studying, what we're really doing is studying the basis of molecular interactions on the scale of proteins, okay? So you move these traps apart to create a force extension curve, something that looks like this, and then you get this unfolding event where you've unzipped the protein. If you relax the force, you go back down this curve, and at a certain point, the protein will decide to fold itself back up. So there's the refolding profile, and you get basically this expectation where you can change the length 34 nanometers, and you get this kind of expected result. However, another method that you can do is put it right at the threshold of this folding and unfolding event. And basically, it will very rapidly, on the scale of microseconds, fold and unfold. And you measure this by the extension of the molecule as it folds and unfolds. So there you have the unfolded and the native state, just characterized by how much time it's spent in those things. So the right-hand side there. So that represents how much time it's spending in the unfolded versus folded state. But if there's misfolding pathways, then these graphs change, okay? So the key is, for misfolding is the unfolded state. So when you've brought this protein into an unfolded state, some of the ways of characterizing how this misfolding is characterized is by what the frequency of the misfolding is. So the folding rate for the proper fold is about 1 inverse second, and for some of the misfolded states, that rate constant is 15 inverse seconds, 4 inverse seconds, and 0.002 inverse seconds. So you can see immediately what the benefit of this is. You can have this protein folding and unfolding in different environments. You can change the substrate that it's in and kind of deduce, like, is this because of a particular chemical that's in the brain? Is it because of the concentration of the misfolded proteins? And now we have tools to be able to actually test these things and understand them a little bit better. So anti-prion agents, how do they actually affect the folding? So here's iron-3 TMPYP, which binds to this protein and inhibits the formation of the misfolded protein, and cures infected cells. So for example, now you can do these same experiments, but with this anti-prion agent present, and see how that changes the folding and unfolding of these proteins. So regular in black, not folded and most common in blue, and high force unfolding in red. So that is one way that you can investigate the mechanism of anti-prion action. Now, we can also study things like the mechanics of frame shifting. So genetic information goes from DNA through a process to produce messenger RNA to the ribosome in the cell to create the protein. So there's several steps involved in actually decoding DNA to produce proteins. And this code is basically written in 3-unit codons that mRNA can read. So when we're talking about frame shifting, we talk about what happens when the ribosome is actually taking this messenger RNA but misreads it by a single letter or by 2 letters. So rather than reading UUC at the beginning there, it might read something like UCU, right? So instead of having a protein sequence of phenyline, serotonin, serine, arginine, tyrosine, something like this. I'm not good with proteins. Asparagine, all right. A frame shift of 2 codons will get you a completely different protein. So this is one of the common ways that proteins can be malformed, is that the code is read incorrectly. So maybe the start or the stop codon has some error in it that causes problems in the protein. And it does occur spontaneously, but at a low rate. So viruses themselves are using frame shifting deliberately to encode more than one protein into a single mRNA. So this is another, like, you know, we're talking about information density at the nanoscale. There are viruses that exist that have figured this out and know that by frame shifting, you can actually create 2 different proteins with the same DNA sequence. So the virus has a way to regulate the production of these two proteins. But it would be interesting as an antiviral drug to create, you know, something that leverages this effect. That's basically it for the synthetic biology and biological aspects. But before we end today, we'll talk a little bit about the perils of nanotechnology. So one worry is this gray goose scenario where you create nanomachines that disassemble organics, for example. And in doing so, you've basically created something that, if it's self-replicating, can proliferate in a way and just destroy all organics. So if something, say, for example, decided it liked to eat cellulose and the wood and plastic in chairs, and it was able to self-replicate, you get into a scenario where you can imagine that 1 copy spawns 2, and they eat enough material to spawn 4 and 8, and this geometric progression basically gets you a whole bunch of these nanobots very rapidly, and they just disassemble everything in sight. And this is the gray goo scenario. Michael Woodside does not think this is something that we should worry about, but self-replicating organisms is a potential worry for synthetic biology. So one thing to consider is the nanotoxicology of these things. So it's not just a question of what materials are made out of, right? If we're talking about silver, we know that as a metal, silver is basically inert. It does oxidize, so you have to polish it every once in a while to get that nice shiny surface back. But what becomes really interesting is when you shrink down the domain size of the silver until you have, you know, a quantum dot sized particle. And you change basically the catalytic properties, you change a bunch of the properties of the material as you shrink it down. So you've heard maybe now of nanosilver being embedded into clothing as an antimicrobial agent. So the silver itself, the material hasn't changed, it's the geometry of the material that's changed and created properties that give it antimicrobial benefits. But we're introducing these things into consumer products at a very rapid rate. So for example, metal oxide nanoparticles are used in sunscreen, and we don't know exactly how dangerous these things are. We don't know what the long-term effects of having these nanoparticles in our environment will be. So it's good for us in this day and age to be very cautious about what we're producing and what we're selling to people, and especially what we're claiming these things do, right? Until we have a lot of studies that tell us exactly how they interact with things, we can claim that, you know, silver nanoparticles are antimicrobial, but who knows if it becomes a problem of, say, bioaccumulation, where silver nanoparticles aren't metabolized by the body, they aren't excreted by the body, so they stay in the body and they do bad things while they're there. So one of the questions that we have to worry about. Carbon nanotubes, something that comes up all the time in nanotechnology, one of the very basic building blocks on a material level for interesting structures like carbon nanotubes, and we've heard about space elevators and strong carbon nanotube filaments. On the nanoscale, they're very similar to asbestos fibers. But so far as, I mean, even today, you can buy carbon nanotubes, presumably for lab work and stuff like this, but this is the kind of thing that you don't want to be breathing in. So these materials, carbon is a great example because it's in everything from our bodies to wood, but in these carbon nanotube forms, they need specific regulation because there are dangers present when it's in this form versus when it's in another. It's not just a material property anymore. It's not just saying this is carbon and carbon is dangerous because of this, right? This is carbon in the form of carbon nanotubes, which gives it properties that are asbestos-like, that are dangerous in a particular sense. So that's the kind of thing that we need to be worried about when we're talking about nanotoxicology and problems associated with nanotechnology. So here's another great example. Gold nanoparticle diameter size is given as the x-axis, and then association constant for different proteins is given. So when different proteins are in the presence of gold nanoparticles, they'll actually denature the proteins depending on what size nanoparticles are present. And so this is gold. I mean, this is a metal that is commonly considered completely inert, but when you put it into this nanoparticle size, you actually get activation of proteins and a denaturing process just based on the size of the protein alone. We did talk about creating structures that aided mobility in the cell. So nanoparticles that can modulate the lipid membrane structure could also be dangerous because they could introduce or expose the cells to dangers in the environment that they wouldn't normally be exposed to. And there are studies that are coming out more and more recently where nanoparticles themselves, even though the materials are inert, cause complications in pregnancies in mice and things like that. There's a great example of silicon quantum dots that were added to zebrafish in great quantities, and it caused the zebrafish to swim backwards. All right, so we don't know exactly why that happens, and that's why it's important to have some caution. So just to give you an idea of the kind of dangers that nanotoxicology presents, there are diseases associated with basically every tissue in the body that are affected by quantum dots. So you talk about nanoparticle ingestion, aspiration, neurological diseases that are caused by nanoparticle-like structures, everything from the skin to the brain, and everything else. So while we've made great strides in nanotechnology in the past, well, let's say as far back as 1959, there are many questions that are still unclear, and it's not clear how to answer them in a general way. So what's the most important thing to consider? Do we measure nanoparticle quantities by mass? Do we do it by surface area, which is a better kind of understanding of, there's a lot more surface area in silver nanoparticles than there is in a lump of silver of the same mass. How do you evaluate toxicity? And can you really evaluate toxicity in all the conditions that this thing will be sold under? How do the properties change with time, aggregation, metabolism? So silicon quantum dots, the ones that I study, are a good example of a quantum dot that is metabolizable, so your body will harmlessly dissolve the silicon quantum dots if they're in a low enough concentration that they're not causing problems to begin with. Where do the substances end up? So cad selenide is another quantum dot structure. The cadmium itself bioaccumulates in organisms and isn't excreted or metabolized. So then you start talking about cadmium toxicology. Even if the quantum dots themselves were protected with a coating, it could be that that coating deteriorates with time and releases the cadmium into the environment that then ends up bioaccumulation. What is the level of exposure from different environments? Can any aspect of toxicology be predictable from the bulk materials? So if you start with silver and you shrink it down, can you predict what that will be like? Has it more to do with the size of the quantum dot, the shape of the quantum dot, and fundamentally, can you model it? So can we predict these things without actually doing a series of experiments? Can we try a bunch of different quantum dot geometries to figure out which one is most effective at binding to a certain target? So while there are a lot of uncertainties, at least we have identified them. So nanospecific cytotoxicology, different toxicity in different cell types. But of course, at this point, many of these parameters are not sufficiently explored. So at this point in kind of the history of nanotechnology, we're at the point now where we need to have uniform practices and standards for comparative testing. And there need to be international bodies that are on regulatory agencies that decide how to do these things. Now, there's the chance that we overregulate and the nanotechnology kind of field doesn't grow at a rate that's sufficient for it to take off as a consumer product. Or you could underregulate and create dangers to humans in the sense of creating nanoparticles and nanobots that do dangerous things instead of nice things. So we need to strike a balance of regulation where we're not overregulating, but again, we're not underregulating. And I mean, this comes up more commonly when we're talking about genetically modified organisms. So it's a topic maybe for another day, but I just want to draw the analogy that when we talk about genetically modified organisms, there's a lot of scaremongering that happens. Basically, GMOs are often changed to help the yields and reduce the cost of growing these materials. But a lot of people are fearful that in modifying the organism itself, you're actually creating something that is detrimental for humans. So that's why, you know, there's a rise in organic farming and stuff like this. And so the same thing is going to happen in the nanotechnology field, where products may be labeled with, you know, this product contains nanotechnology, and maybe you even need to devise a little symbol that says, you know, danger, nanoparticles present, and maybe an explanation of what that particular danger is, like do not ingest this ink, for example, that has nanoparticles present. And I guess most fundamentally is, how do you distinguish nanotechnology from conventional biology? So if you're producing a bacteria that creates a biofuel, at what point do you say this is no longer a bacteria that produces a biofuel, this is a genetically engineered organism that produces a biofuel? Because standard genetic techniques lead us to bacteria that can do these things. That's before we have any nanotechnology. And so there needs to be a line drawn between conventional kind of chemical and genetic techniques and that which we consider nanotechnology. Or maybe there should be no division, and it needs to be just all-encompassing in terms of, you know, molecular regulation. So the risks of nanotechnology must be framed appropriately. Here's a picture of some alligators on the side of a road saying, hikers and bikers move to the side of the road when the vehicle approaches. So is it really prudent for a hiker or a biker to step out of the path of a moving car and into the jaws of an alligator? So frame our risks appropriately. A lot of the material that was in this lecture came from Steve Block at Stanford University, Robert Wolko at the University of Alberta, Laurie Sharametta at the University of Alberta, and Paul Burrows at Riata Research. And this lecture was originally by Michael Woodside at the National Institute for Nanotechnology in Edmonton, Canada. So I think that we covered kind of, you know, a pretty diverse background in terms of what you can do with nanotechnology and kind of what the state of the art is. What should be mentioned is that this probably hasn't changed since 2010, so there's maybe 5 years worth of research that can be added to this in the meantime, and that being said, there's all sorts of cool stuff that has happened in the meantime.
Yeah, it's sort of fascinating. If you go to the YouTube channel for the course and look at the most popular lectures, they include one by Jonathan Schaefer and by Michael Woodside, and they're both approaching 3,000 views. But both of the individuals who did those lectures consider them so out of date that they're badly in need of updating. So some of the most forward-looking and precise and, you know, modern lectures there have very few views, whereas some of the ones, Jonathan Schaefer tells me that when he steps down as dean, his first act is going to be to bring that lecture up to the modern era. So, yeah, one repeated discussion point has always been from the beginning here, and I can give you what Michael Woodside's answer was. What about regulating nanotech? You know, like I'm in a field that's heavily regulated. You know, people get peer review and all sorts of things, and if you're regarded as kind of an evil person, you somehow don't make it into medicine. With nanotech, there's nothing like that. And so I used to ask Michael whether that means that naturally good people just gravitate into the field. So we should be able to trust these people because they're good scientists and all this kind of thing. And he didn't feel that that was really the case at all. It's sort of a random sampling of bright human beings. So the danger is hard to define because, just as Ross has been saying, you know, it's hard to say where things begin. When exactly does dangerous nanotech begin, and you know, how do you define it? It's not without dangers, but it's hard to figure out. Like, let's take your suntan lotion and, you know, the stained glass windows in your church, you know, that sort of thing. Are all those things dangerous? Well, probably not. But, you know, they contain nanoparticles. So the book Prey, I don't know how many of you have read that book, but it is quite scary, and that it was a very popular book, and that, you know, is sort of.
There's another one, maybe you've read it, but Diamond Age?
No.
By Neil Stevenson.
Yeah.
Diamond Age is one of my more favorite kind of future nanotech apocalypse scenarios, and basically they've got to the point there where, you know, particles of dust are doing computations and stuff, and actually the entire world is permeated by like nanobots that you can see their little searchlights as they float through the air. But they're waging a war during the novel, and the main character is basically embroiled in how those nanoparticles are affecting the events of the plot, more or less, without spoiling too much.
You mentioned that you use the DNA origami techniques to create a lockable box for the drug delivery.
Yeah.
How does this box break down, or can they control the degradation rate of this box for the purpose of for the drug delivery?
Yeah. So when you've got the lockable DNA box, what happens to it when it's open, basically in the body? Is that the question?
Yeah.
Yeah. So more or less, when you're using biologically compatible structures like DNA, the body will identify it as something that it can be broken down into its constituents and reused in the cell itself. So while I don't know the specific fate of the box structures and the DNA origami structures, it is easy enough for me to imagine that just gets metabolized back into the body through natural processes. And that's kind of one of the benefits of doing that kind of synthetic biology, where you're starting with something that's already biologically compatible with humans to introduce kind of, you know, this more specialized drug delivery aspects. Okay. But at the same time, so like a silicon quantum dot, for example, for the most part, they're inert and they're readily oxidized. So there's breakdown pathways for silicon quantum dots that basically turn it into, well, more or less turn it into sand, but it gets broken down into even smaller pieces that are eventually excreted from the body as well.
Okay, thanks.
Yep.
So I have a question that doesn't relate to the bacteria, but rather my question is more not so much medically related, but you know, we've always had the concept, it's been more sci-fi than anything, of you know, stuff that can repair itself, of, you know, you break it and it fixes itself. And a lot of, I guess, what I've read is that to really achieve that point where we get objects, you know, daily objects that can repair themselves, is we need advances in nanotechnology to really achieve this. And I guess what I'm asking is this really a feasible thing that could happen, in which, and I don't mean like, you know, really, really far away, but like in the reasonably near future, could we get, you know, maybe not everything, but at least a lot of expensive things like electronics, laptops, TVs or whatnot that, you know, get dropped once and they're gone forever. Could they be made to replace themselves with advancements in nanotech?
So there's kind of two things that come to my mind to answer this question. The first is that we're talking about repairability, but on the one hand, so that's the question that I want to answer is, can things be repaired on the nanoscale? On the other hand, something you touched on is more about the durability of things. Like when we drop our phones, they tend to shatter and break. So on the one hand, like the reason that we have phones is because of nanotechnology in terms of the battery technology, the processing technology, but even things as innocuous as the glass technology. So Gorilla Glass is a really great example of a structured, so think geometry, a structured material that is stronger than its constituents in a sense that if they made it purely out of glass, it would have material properties that wouldn't be suitable for a phone. But if they made it purely out of plastic, it would also have material properties that wouldn't be suitable for a phone. So by mixing and laminating and creating these new structures, you're actually producing something that has the benefits of both. And that, I think, is very early in its nascence, that we're going to be seeing materials that are far more durable than the ones we have today, based on things like embedding carbon nanotubes into support structures. Making electronics smaller will obviously make them more difficult to break, right? If your processor is you know, the size it is today, like the main board of your smartphone is the size of the smartphone, and you break it, that's it. But if you shrink the main board size down to a grain of sand, conceivable, maybe not in the next 10 years, but if you shrink it down, you're reducing the overall ability for that object to break. So that's one sense in which I think that nanotechnology is going to drive the durability of objects through the roof. And at that point, I'm thinking like, you know, you might even buy a phone, and then you might decide that your phone is too big, and you might snap off, you know, an inch of it, and then the new size structure just works as a phone. Like that's the kind of technology that we're driving towards, where we have like these universal screen sizes, very strong materials, and it could be that eventually, you know, this is something you buy for your 3D printing kit at home. You print a screen, and then you plunk in a little screen module, and you know, this thing lights up. Now, we're kind of in the sense where we're in the 1950s for nanotechnology, the way Richard Feynman was in terms of electronics. And so when I say, you know, we'll 3D print a screen, the only thing I can imagine is a conventional 3D printer that we have today printing, you know, squirting out some plastic. But that's the kind of thing that you get stuck into you get shoehorned into this idea that 3D printing must be a specific thing, when really there's nothing stopping a 3D printer from doing something like having electronics embedded in the actual filament that gets extruded, or embedding it in it during the process, or having a filament that's made out of something that is, you know, materially complex. So don't discount the idea that these innovations will come from outside of the box, from things that we haven't experienced yet. And you know, that's the kind of thing that gets really surprising, that you go, oh my God, I can't believe you could do that, whereas, you know, yesterday you couldn't, today you can. Now, what are all the cool things that you can do with this, right? So a company that I'm following quite closely is called Color Fab. They produce 3D printed filament, and they're doing things like glow-in-the-dark filaments, and just recently they have a carbon fiber filament, so you can actually print PLA plastic with embedded carbon fiber for greater strength. And it's that kind of innovation that you really can't predict where that will go. One thing that I would love to do is to just do some very simple optical properties of 3D printed materials. So can I produce an optical isolator by having an optical element on this end, an optical element on this end, and then 3D printed optical traces between them? It's a very simple first step, but that's the kind of thing, you know, like I could walk down to the 3D printer, I've got transparent filament, I can create an optically active 3D print. The question is, you know, this is the dot matrix of 3D printers. When we've got the equivalent of laserjet 3D printers, and they can do different materials and different conductive properties and different optical properties, you know, that opens up the whole ballpark of new materials. Now, as far as repairability goes, that's the more complex part, because then you need, you know, a nano machine that is designed for a specific job. And you have to realize, okay, I'm going to get the analogy wrong, but let's stick with the analogy for today, and if I'm very wrong, I'll correct myself on Thursday. We're on the human scale here, and the universe is huge, right? The universe is 10 to the 80 atoms or something crazy like that, and we're just kind of, you know, we're medium scale as far as the size of the universe. But what people forget is that for the vastness from human scale to cosmological scales, that vastness also exists from human scales to the nanoscale. So where we're talking about, you know, we drive cars and create spaceships that can fly through space, when you're talking about the really small end of the nanoscale, when you're working with atoms, you're creating rocket ships for the universe that is the nanoscale. That when you're creating these machines, they're operating in a universe that is smaller than us, but nonetheless as big as the one that we're trying to operate in. Because humans, you know, we can't make cars that are smaller than human to transport humans, right? There's a limit at the moment for how big a person is, right? And it fits in between, you know, the smallest human and the biggest human. And so we say, well, we can only make rocket ships that will carry humans, you know, and so the rocket has to be bigger than the human. But on the nanoscale, you're not limited by this thing. So for every vehicle that you can imagine, cars, trucks, rockets, if you're constructing them out of atoms, you know, they can be big on atomic scales. They can be 100 atoms across or 1,000 atoms across and still fit through your bloodstream. So while it's hard to imagine at the moment, things like the replicator that was on the first slide, basically like a little biological robot that is sitting with red blood cells in the body, like those aren't outside of the realm of reality. It's just that the building blocks necessary to create them aren't robust enough. And that's one of the problems, is that should you be able to create one of these, great, you've done a fantastic job. The problem isn't creating one. The problem is creating enough to make an effect, right? Even if there was one of these in my body right now, would my body actually see a difference? Because that one might be able to repair, you know, 1 red blood cell per second. But if I've got leukemia, then the rate at which my blood cells are, I don't know enough about leukemia, but yeah, the number of blood cells that there are to fix can't be held by just one of these. So the problem at the nanoscale is not simply making something, but making enough of that something to make a difference on the human scale. And that's where I think, you know, we talk about manufacturing things on the nanoscale, and we're getting better and better and better and better. There will come a time where, you know, someone's awarded the Nobel Prize, or someone becomes tremendously rich overnight because they say, look at this cool thing that I've done. I've taken an existing structure and I've made 1 trillion copies of it. And that's huge. So that's where we're stuck on the repairability. You may be able to create the devices that recognize that, oh, this carbon's not in the right place, or this protein misfolded, and I can change it by, you know, putting my pincers up in this way, right? When we're talking about intelligence and machine intelligence, it's hard to, let's not go too far into that direction, but while it's conceivable, the real problem is still just copying things. 3:20, I think that's it. All right, see you guys on Thursday with a more well-rehearsed and prepared lecture.
Thank you very much.